Deep inside our bodies, there exists a microscopic world of incredible complexity. These filaments are called actin and myosin, and without them we wouldn't be able to walk, talk, or even breathe. Imagine a team of rowers pulling in perfect unison, their oars propelling the boat forward. Actin forms the long, thin filaments that act like the boat itself, while myosin forms the oars, pulling on the actin filaments to create movement. This interaction between actin and myosin is what allows our muscles to contract and generate force. From the beating of our hearts to the digestion of our food, actin and myosin are involved in countless processes essential for life. Understanding how actin and myosin work together to power our movements is like unlocking a fundamental secret of life. Not all muscles are created equal. Our bodies rely on three distinct types of muscle tissue: smooth muscle, cardiac muscle, and skeletal muscle. Smooth muscle is responsible for the slow, sustained contractions in our internal organs. It lines the walls of our digestive system, blood vessels, and airways, working tirelessly behind the scenes. Cardiac muscle, found exclusively in the heart, is responsible for the rhythmic contractions that pump blood throughout our bodies. Skeletal muscle is responsible for the voluntary movements of our bodies. Each type of muscle plays a crucial role in our survival, powered by the interplay of actin and myosin. To truly appreciate muscle contraction, we need to delve deeper into the structure of muscle fibers. These fibers are like long cylindrical threads, bundled together to form the muscles we see and feel. Within each muscle fiber, we find myofibrils, the contractile units of the muscle. These rods are further divided into segments called sarcomeres, the basic functional units of muscle contraction. Surrounding the myofibrils is a network of interconnected sacs and tubes called the sarcoplasmic reticulum. This network acts as a storage unit for calcium ions, essential messengers that play a critical role in triggering muscle contractions. The sarcoplasmic reticulum ensures that calcium ions are readily available when needed. The structure of muscle fibers highlights the incredible complexity and efficiency of our muscles. Section 4, the actin-myosin tango, a dance of contraction. Let's bring our attention to the heart of muscle contraction, the interaction between actin and myosin filaments within the sarcomere. Picture two rows of interlaced protein filaments, thin actin filaments and thicker myosin filaments. When a muscle receives the signal to contract, the sarcoplasmic reticulum releases its stores of calcium ions. These calcium ions bind to the actin filaments, causing a change in their shape. This shape change exposes binding sites on the actin filaments, allowing the myosin filaments to attach, forming cross bridges. Once attached, the myosin filaments perform a kind of molecular dance, pulling the actin filaments closer together. This sliding of the actin filaments past the myosin filaments causes the sarcomere to shorten. As millions of sarcomeres shorten simultaneously, the entire muscle fiber contracts, generating force and causing movement. Section 5. Actin and myosin, the unsung heroes of movement. Actin and myosin are not just proteins, they are the fundamental building blocks of movement. Without these tiny protein filaments working tirelessly within our cells, our bodies would be incapable of coordinated movement. Their interaction within the sarcomere is a testament to the power of simple yet elegant design. Our understanding of actin and myosin has far-reaching implications for developing new treatments for muscle diseases and injuries. By unraveling the secrets of these microscopic engines, we gain a deeper appreciation for the wonders of the natural world and open up new avenues for improving human health and well-being.